Hello and welcome to Botany 101. My name is Angela Nishimoto. Okay, as you recall, last time we were talking about true bacteria, their forms, some of their functions. Okay, so today we're going to continue with bacteria, archaea, and viruses, part 2. All right, so for our outline of topics, we're talking about bacteria, continuing with bacteria, and we're going to talk about blue-green bacteria and etc., other kind of bacteria. We're also going to talk about the archaeans, and we're going to talk about viruses. Okay, so first of all, we're going to talk about the true bacteria useful to humans. Okay, so for decades we've been relying on chemical insecticides to control pests on crops, right? So there's some problems with that, in that, for one thing, the residues from these sprays have accumulated on fruits and vegetables, and we consume them and they accumulate in human tissue. That's one bad thing. Another bad thing about it is that insects over the decades have become resistant to these toxins. Okay, so they don't die quite so readily as they used to before. Okay, and thirdly, these insecticides also kill insects that they are not meant to kill. Okay, and also the runoff from these insecticides go into streams and rivers and lakes and ponds and oceans, and they can harm aquatic life at the same time. So we've been looking for alternatives to these insecticides. Okay, so what we're looking at now is at biological control on pests. Okay, we talked about Bt before. It comes from a bacteria called Bacillus thuringiensis. Okay, there's another related or in the same genus bacteria called Bacillus papilii, so we'll be talking about these. Okay, so Bt is very effective, okay, on caterpillars and worms. All right, that's very good. All right, so Bt as an insecticide is sold as a wettable dust, and this wettable dust has millions of spores in it. Okay, so they're harmless to all insects except moth and butterfly larvae. Okay, so caterpillars and cutworms will eat the spores that would be on the leaves of the plants, and the bacteria becomes active and multiply in the gut. Okay, and the caterpillar stops eating in about 2 to 3 hours, then dies and turns black. Okay, within 2 to 3 days. Okay, so Bt is very deadly to caterpillars and cutworms. Okay, so the gene for Bt has been introduced into crop plants, where the plants actually produce their own Bt, and these include corn crops and also soybeans. Okay, and this Bt produced internally by these transgenic plants are especially useful for internal insects like cutworms, and cutworms are actually in the internal leaves and internal organs of the plant, so they're not affected by sprays. Okay, so in 1983, a variety of B. thuringiensis called variety israelensis was actually introduced to control mosquito larvae. They're also known as wigglers. Okay, and here's a mosquito larva here, so many of you may be familiar with it. This is the B. thuringiensis, variety israelensis. Okay, here's an adult mosquito. Okay, so what they do is they'll kill the mosquito larvae. Okay, and apparently they have no real bad effect on plants or animals. Okay, they've done 6 years of experiments on fish and snails and shrimps and insects, showing that they have no adverse effect even at 100 times the concentration needed to control the wigglers. Okay, so no bad effects on plants or animals except mosquitoes and a few others that are related to them. Okay, So Bacillus papilii is applied to soils, and it kills grubs of the Japanese beetles. Now, Japanese beetles are extremely destructive pests. Okay, they eat practically anything, fruits, leaves, etc. So what happens is that this bee papilii applied to soils will kill the grubs. They come down with something called milky spore disease, and they'll die within a few days. So all they have to do is dust this bee papilii dust on the soils, and water plus other insects, and just the movement of the soil will actually carry it to the grubs that live in the soils. Okay. Now, bioremediation is another use of bacteria to humans. Okay, bioremediation is the use of living things to clean up toxic wastes and pollution. Okay, so bacteria can break down nitroglycerin and trinitrotoluene, and what these are contaminants around munitions factories and explosion sites. Okay, they're in the soils. So the bacteria that they can use for these can decompose them within about 6 months. Okay. Now another bacterium called Pseudomonas cepacia, okay, can clean up oil spills and chemical dumps containing degreasers, such as trichloroethylene or TCE, creosote, which is the smelly part in telephone poles that makes telephone poles, you can't really use them for anything else. You must dispose of them after they're no longer useful. Okay, they're used as anti-termite. And the famous 2,4,5-T, an aspect of Agent Orange. Okay. So some also believe that bacteria can be used to break down nuclear wastes, okay, in the future. Okay. That's pretty important, right, in our modern day. Now, other useful bacteria, some have a form of rhodopsin, which is a protein that's sensitive to light. It's been very valuable in research on the eye, because of course our eyes also have rhodopsin in it too. Okay. And bacteria play a major role in dairy industry. Okay, so except for milk alone or mixtures such as ice cream. So what is milk? Okay, milk is proteins, carbohydrates, fats, okay, vitamins and minerals, okay, about 87% water, so it's very nutritious for mammals. It also makes a really good nutrition for bacteria as well, okay. So except for milk alone or mixtures such as ice cream, all products that are dairy are made from the controlled introduction of different bacteria, okay. All these products, buttermilk, acidophilus milk, yogurt, sour cream, kefir, and cheese. Okay, cheeses are all made by using bacteria, okay, important in the dairy industry. Now, whey is also used. Whey is actually the watery part of milk that's separated from the curds during cheese making. So whey is the watery part, and whey is used along with starches and molasses to produce lactic acid. Okay, and lactic acid is used in producing textiles, laundry products, and it's used in leather tanning. It's used as a solvent in lacquers. It's used also for production of supplements for the treatment of calcium and iron deficiencies. Okay. And Lactobacillus acidophilus, a lot of you might have heard about this. This is a bacterium that is very common in healthy digestive tracts. Okay, and this Lactobacillus acidophilus, if you don't have enough of your own, you can go to the health food store and buy a mixture in a pretty good tasting formulation that you can take, like a tablespoon or two of every day. Okay, so this aids in digestion. All right, It's supposed to also decrease the risk of certain cancers. Okay, the presence of Lactobacillus acidophilus. Okay, it's supposed to also decrease cholesterol levels. It's supposed to help decrease cholesterol levels, and also control yeast infections in females. Okay. So the bacteria also produce products that are important in industry. They're oftentimes produced in large amounts by culturing bacteria in these huge vats. Okay, so acetone is a product made from bacteria. Okay, explosives shown here. This is acetone. This is some explosives, solvents. Okay. Butyl alcohol, which is used to make lacquers. Okay. Dextran, which is a food stabilizer. It's also a blood plasma substitute. Okay. A substance called sorbose, which is used to make ascorbic acid or vitamin C, and also citric acid, which lends its kind of tart taste to sodas. You shouldn't drink sodas actually, but it lends its tart taste to sodas, candies, gum. Okay. Also some vitamins and minerals also are products that are produced, okay, by bacteria. Okay, bacteria also cure vanilla pods. Okay, cocoa beans, here's some fruit on the chocolate tree. Coffee to ferment it, and black tea to ferment it. Okay. Also, bacteria are used to produce vinegar. Vinegar comes from the souring of wines, right? And sauerkraut, which is very good for you, because these fermented vegetables are very good for you, all right? Good for the digestive tract, if your digestive tract can take it. And dill pickles, also another product. Linen fibers are separated from their flax stems using bacteria. Green plant materials are fermented for silage, which is actually made in silos. Okay, so the green plant materials are stored in silos, fermented for silage, which is fed to cattle and other livestock. Okay. Bacteria also are used in the production of amino acids, such as, let's see, 7,500 tons of glutamic acid are produced every year in the United States. That's used to make a flavor enhancer called MSG, or monosodium glutamate. Some people are allergic to monosodium glutamate, though. Okay, and this bacterium called Brevibacterium is used to make Limburger and Brie cheeses, all those nice smelly cheeses that people like so much. Okay. So what we're going to do now is we're going to go take a look at some cultures of bacteria here at the side table. Okay, so we have two different types of bacteria here. Okay, we have Bacillus subtilis, which of course is a bacillus bacteria or rod-shaped bacteria. Okay, so we have two cultured petri dishes full of these, and we have these little marks from somebody making the loop marks with it to culture it some more. Okay, so this Bacillus subtilis or B subtilis is actually a component of the normal gut flora, right? And this is Micrococcus luteus, okay? Micrococcus luteus, which is cocci bacteria, right? So a cocci bacterium is spherical, right? And so this particular bacterium is actually part of the component of the flora of on your skin, okay? But these are pure cultures. You can see they have on the petri dishes do not open, okay, because you don't want to be messing with bacteria, but I just wanted to show you these cultures. So we're of course not going to open them because they're don't want to get contaminated, as well as you probably do not want to be exposed to these bacteria. They're not necessarily dangerous, but there are some strains of some bacteria that are that are not dangerous that actually can cause disease, such as MRSA, which is that methicillin-resistant Streptococcus aureus. Okay. Anyway, you can see from these colonies that you can tell that they're different bacteria. Just wanted to show you that. Another thing I wanted to show you about this was that they're cultured on agar. See this kind of yellowish, kind of jelly-like substance in these petri dishes. Okay, we'll be talking about agar more in the future. Okay, that's all I wanted to show you here. Now we're going to go back to the computer. Okay, so here we are. Okay, now we're going to change our pace a little bit and get into a different class of true bacteria called the cyanobacteria. Okay, they're also known as the cyanobacteria or blue-green bacteria. Okay, they used to be called blue-green algae before because they do photosynthesis. Okay, but while true algae have eukaryotic cells, the cyanobacteria, of course, have prokaryotic cells. Okay, they also contain chlorophyll A. Okay, which of course is a form of chlorophyll, a plant pigment. Okay, and they produce oxygen gas during photosynthesis. Okay, whereas the other photosynthetic bacteria we talked about a little earlier do not produce oxygen gas. Okay, other pigments they may have or do have are the phycobilins. Phyco means alga, so these are known as the blue-green algae before, even though they're more truly bacteria. But phycobilin pigments they have, the blue phycocyanins and the red phycoerythrins. Excuse me. And they also fix nitrogen, some of them, and produce oxygen gas. And they're the only organisms except for the prochlorobacteria, which we'll be talking about later, okay, that do these. Okay, the other bacteria which do photosynthesis do not produce oxygen gas, and they also do not have chlorophyll A. Okay, so here's a look at a cyanobacterium. Okay, here we have the cell membrane. Okay, here you have the peptidoglycan layer. This is where the muramic acid is in the peptidoglycan. Okay, you have an outer membrane. This one has a mucoid sheath like mucus and a slime coat. Okay, charming. Anyway, so you can see the ribosomes, the nucleoids. So this is really a bacterium. Okay, and it has rubisco. Remember, that's a particular enzyme used in photosynthesis. Okay. So with their distribution, where are they found? They're especially found in temporary pools and ditches, especially if the water in the temporary pools and ditches is polluted. Okay. But they're not found in acidic waters. Okay, but they're abundant in other freshwater and saltwater environments. Okay, from the frozen lakes of Antarctica all the way to the warm tropical seas. All right. And in open oceans, they're the main photosynthesizing organism in the plankton, in the phytoplankton. Okay, the phytoplankton are microscopic organisms that do photosynthesis. Okay, whereas the zooplankton actually eat phytoplankton. They're like little animals. Okay. Now, Synechococcus, it's been estimated there are about 10,000 cells per milliliter of seawater. Okay, so that's actually 1 milliliter is 1 cc or 1 cubic centimeter. So you know how small that is. Yeah, so there are 10,000 cells approximately in 1 milliliter. Okay, here's a look at the ocean. Okay, with some, looks like some blue-green bacteria here. Okay. Now, Trichodesmium is a marine and filamentous bacterium, and it also does nitrogen fixation. It forms extensive mucilage-producing colonies in tropical waters. Okay. Now other bacteria use the mucilage to multiply in, and these other bacteria become food for protozoans. Okay. Now in Yellowstone National Park, there's a different blue-green bacteria found in each temperature range in the springs around Yellowstone. Okay. Blue-green bacteria in Hawaii are among the first photosynthetic agents on bare lava. After the eruption, they actually colonized bare lava and they start to break it down. Okay. They also thrive in desert rocks. They live in soils, on shells of turtles and snails. And there are others that are symbiotic. That means they live with other organisms, such as amoebas, diatoms. Remember we talked about diatoms before, or we will be talking about diatoms later on too. Okay. Sea anemones, which are animals. Fungi, they live symbiotically with fungi. Okay. They also live among the roots of these gymnosperms, or plants that bear seeds, but in open structures called cycads. Okay. They're also components of lichens. So here's some sea anemones in this nice artist rendition. And here's a lichen. This looks like a foliose lichen. Okay. Lichens are actually made up of a fungal partner, and either a blue-green bacterium or a green alga. Okay. The form, metabolism, and reproduction. Okay. Often they occur in chains or in hair-like filaments. Sometimes these filaments are branched. Okay. They can be in irregular, spherical, or plate-like colonies, with the individual cells held together by gelatinous sheaths. Okay. So for about half of the 1,500 species of blue-green bacteria that are known, the cells tend to be blue-green. Okay. The pigments from green chlorophyll A and blue phycocyanin. Remember we talked about phycocyanin a little bit before, also about phycoerythrin, right? We'll talk about that more in a sec. Okay, so carotenoids and phycoerysthrins are sometimes present. So the blue-green color can sometimes be masked by these pigments. Okay, so that would create a red color to the blue-green bacteria, which sounds kind of funny. But the Red Sea is actually called the Red Sea because periodically there are blooms of blue-green bacteria in it, which colors the sea a red color. Okay. Now cyanophycin is a nitrogenous food reserve. So it's unusual for bacteria to have food reserves, but they have a nitrogenous food reserve called cyanophycin. And they also produce and store carbohydrates and lipids as also food reserves. Okay. How they reproduce. Okay, they actually reproduce through fission, okay, to make new cells, and they also reproduce through fragmentation, where you get new colonies or filaments from fragments of an old colony or filament. Okay, now flagella are unknown in cyanobacterium, but some are still capable of movement. Okay, they have like an oscillating movement. Some of them do, very few of them oscillatorium among them. Okay, now Nostoc and Anabina are common genera. Okay, they form chains of cells, as you can see here. You can see the green pigments in here. Okay, Nostoc and Anabina. Okay, these can actually do fragmentation at these cells called heterocysts. So heterocysts are sort of larger, sort of colorless cells, and they fix nitrogen, so this might be a heterocyst here. Okay. You can also have thick-walled aconites. Okay, and these aconites actually can resist freezing. They can resist other adverse conditions like drying. And there's actually been like a record of maybe 80 years where an aconite has actually been in its dormant state, and then it germinates and continues to grow after 80 years. Okay, so like the other true bacteria, they do not produce gametes or zygotes. They also do not do meiosis, okay, or mitosis. Okay, now cyanobacteria, chloroplasts, and oxygen. Okay, now symbiotic cyanobacteria often lack cell walls. Okay, so these look like plant cell walls here, probably of Anacris or Elodia. Okay, so these symbiotic cyanobacteria, they live in symbiosis with other organisms, and they function as chloroplasts in these organisms. Okay, so chloroplasts probably originated, the organelles, the chloroplasts in green plants, originated as cyanobacteria or prochlorobacteria. Okay, and these ended up living in other cells. Okay, so symbiotic cyanobacteria do function as chloroplasts in organisms they live in. Okay. Now, about 3.5 billion years old fossils, okay, from Australia. Okay, they have these fossils. They look like cyanobacteria, like modern day cyanobacteria. Okay, 500,000 years later, they produce oxygen gas as a photosynthetic byproduct. Okay, and in the atmosphere, oxygen gas accumulated. Okay, then other photosynthesizing organisms appeared, like the algae. All right, and what happened after that? Aerobic respiration developed. Okay, because oxygen gas was actually the world's first air pollution. Okay, there wasn't that much oxygen in early Earth atmosphere. Okay, so aerobic respiration developed within the last 500 million years, and that accumulated enough ozone to shield the Earth and the living things against too much ultraviolet radiation. Okay, so ozone is O3, which is a byproduct of O2, oxygen gas. Okay, so think of the implications of this. Okay, because there was enough ozone accumulated to shield against UV radiation, the photosynthetic organisms could now survive on land. Okay, and when the photosynthetic organisms, the plants or the algae, became land plants, you had animals that ate the plants following them onto land, and you had other animals that ate those animals following them onto land. Okay, so therefore, the cyanobacteria has played a role in the history of all living things. Okay. Now, human relevance of cyanobacteria. They are the beginning of food chains. They store energy and sugars from photosynthesis, like all producers do. Okay. Small fish and crustaceans eat these cyanobacteria. They in turn are eaten by larger fish, and the larger fish, of course, serve as food for other organisms, including humans. Okay. And sometimes you'll have a floating scum or a bloom of cyanobacteria. Okay, this usually happens during warm weather, like in late summer in temperate areas. Okay, now these blooms become abundant when you have a lot of agricultural runoff high in nitrogen and phosphorus. Okay, not just that, as the water starts to smell fishy from the cyanobacteria in them. Okay. So Anabina, Microcystis, and others. So here's Anabina. Here's Microcystis. Okay, they can produce toxins, and these can kill animals. Okay, so these animals include cattle, pigs, horses, sheep, dogs, even poultry, like chickens and geese and turkeys. Okay, rabbits also die sometimes when they've taken in toxins made by these cyanobacteria. Okay, and cyanobacteria can be attacked by cyanophages. Okay, cyanophages are viruses that And there's some interest in using these to control blooms, okay? But many of the blue-green bacteria that cause blooms actually have a mucilaginous sheath, okay, that prevent the viruses, also fungi from entering the cells. So that's kind of not really useful. And there was a bloom off Australia once that was measured at about 1,000 miles long and covered 20,000 square miles. So here's actually a photograph from the International Space Station showing cyanobacteria. You can see this kind of bluish green color here. It's a bloom of cyanobacteria. Okay, this is Tasmania. Okay, I guess this is Australia here. Okay, so you can actually see that from space, amazingly, huh? Now, not many of them are used for food because a lot of them are smelly and some of them are toxic, but spirulina has been used for food by people near Lake Chad in Africa and the Mexico City areas, okay, around Mexico City for probably thousands of years, hundreds of years at least, okay? And now spirulina, of course, is cultured commercially. You can get them in health food stores. Okay, it's supposed to be very nutritious for you. They have a lot of vitamins in them. Okay. Now some nostoc strains produce antibiotics that kill related strains of nostoc. Okay. So Cytonema hoffmanii also produce cyanobacterians, which are antibiotics that kill cyanobacteria and algae. So these maybe can be useful to us if this is what we want. And swimmer's itch in Hawaii, you know, sometimes people get like a severe case of itching from this blue-green bacteria in the waters off Hawaii, called Rhylingbia. Okay. Now the interesting thing about that is that the toxin that's produced by Rhylingbia actually suppresses leukemia and other cancers. Okay. Now about the water supplies. Cyanobacteria tends to clog filters. They also tend to corrode steel and concrete. They can cause softening of water. They can produce offensive odors and bad coloration that you don't want to see in the water. You don't want to swim in water that has cyanobacteria in it because it kind of looks kind of yucky. Okay. Now, cyanobacteria can be controlled in water supplies by the application of very, very dilute solutions of copper sulfate. Okay, now more than 40 species fix nitrogen of cyanobacteria. Okay, they fix nitrogen from the air at about the same rates as nitrogen fixing bacteria of legumes, which we talked about before. Okay. In Southeast Asia, there's so much nitrogen produced in rice paddies by these cyanobacteria in these ferns called azala that they can grow rice for years without adding fertilizers. Okay. Now we're going to start with the class Prochlorobacteriae, okay, or the Prochlorobacteria. So in 1976, Ralph Lewin of the Scripps Institute, okay, of Oceanography, published about these unicellular prokaryotes with bright green cells that were living on sea squirts off Baja California. Okay? So we call these cells prochloron. All right? So they have chlorophylls A and B, and also carotenoids, but they do not have phycobilins. Okay? Furthermore, they have a double membrane, unlike the single membrane of cyanobacteria. Okay? And in 1984, Dutch biologists discovered Prochlorothrix, excuse me, in the Netherlands. It lives in shallow lakes. And these particular Prochlorobacteria are free living in filamentous. Okay, whereas the earlier one discovered lived in sea squirts. They were symbiotic with sea squirts. Okay. Now in the 1980s, Sally Chisholm of MIT discovered another prochlorobacterium. So I guess they call it Prochlorococcus. Okay, so this one actually lived in dim light at about 330-foot depths. Okay, and now they think it is one of the two most numerous bacteria in the oceans. Isn't that amazing? Okay, now we're going to get into a different domain. So we're done with the bacteria, okay, for now. Okay, so we're going to go ahead and get into the domain or kingdom Archaea, the archaebacteria. Okay, so they differ from true bacteria in their metabolism, completely different. Okay, they have different sequences of RNA bases. Okay, they have a lack of muramic acid, no peptidoglycan in their cell walls, and they also produce distinctive lipids. This is actually muramic acid. Okay. So in the 1980s, Carl Woese of the University of Illinois thought they should be in a different kingdom than bacteria. Okay. But later workers actually thought that the differences between the archaebacteria or the archaeans and the bacteria are so fundamental that they should be elevated into separate domains. Remember that the domain is actually a larger grouping than the kingdom is. Okay? So we're going to do a survey of the different kind of archaea. First of all, we have the methane bacteria. So these are most of the archaebacteria. Okay? They are killed by oxygen gas. Okay? They're active under anaerobic conditions. So they live in swamps, ocean and lake sediments, in hot springs. Okay? They live in the intestinal tracts of animals. And they live in sewage treatment plants and other places like that have little oxygen or no oxygen. Okay? So they get their energy for the generation of methane from carbon dioxide and from hydrogen. Okay? Look at that flame there. So methane is a principal component of natural gas. Okay? And it was a major part of Earth's early atmosphere, and it's still present in the atmospheres of Jupiter, Saturn, Uranus, and Neptune. Now methane burns when it's only 5 to 6% of the air. Okay, so the dancing light, the ignis fatuus, or the will-o'-the-wisp, okay, tends to be seen at night over swamps and marshes. Okay, people think it's a spontaneous combustion of methane, okay, in the air above swamps and marshes. Okay. Now the second group, the salt bacteria, thrive in extreme salinity. Okay, and this salinity will kill all other cells except the salt bacteria immediately. Okay. The salt bacteria do photosynthesis with bacterial rhodopsin, which is a red pigment. Okay. So the salt concentration in the cells tend to be lower than in the environment, but these cells will die if they're placed in water with lower salt concentrations. Okay. So commercial salt ponds and bodies of water with high salt content oftentimes are red. Okay. From the pigment, the bacterial rhodopsin, which is a red pigment. This year is actually part of the Great Salt Lake. Okay. I think this is supposed to be Antelope Island, somebody told me. Okay. So here's a look at the Great Salt Lake, and you can see that this area here is pigmented red from the salt bacteria. Okay. Of course, the Great Salt Lake and the Dead Sea are very high in salt concentrations. There's supposed to be some kind of a railway line here or something that demarks one side of the lake from the other. Okay. Now the last bacteria we're going to talk about in terms of the archaebacteria are the sulfolobus bacteria. Okay, they occur in sulfur hot springs. So look at that. and they thrive at high temperatures of like about 176 degrees Fahrenheit. Now this bacterium called Pyrodictium has a minimum temperature requirement of 179 degrees Fahrenheit, and it has optimal growth temperature at 220 degrees Fahrenheit. It can actually stand temperatures of 230 degrees Fahrenheit. Okay. Now the environment of one order is also acidic with a pH of less than 2. And this other organism, thermoplasma, has no cell wall. This is actually a picture of it. Here's a little scale to show how big or how little it is. Okay. And this thermoplasma is found only in the embers of coal tailings. Okay, so it's actually thermophilic. So Thermoproteus is found in geothermal areas of Iceland. Okay. Now James Lake and other people discovered that the ribosome shape of sulfolobust bacteria are different than the ribosome shapes of the ribosomes in archaebacteria, true bacteria, and the eukaryotes. Okay. They also have a different kind of chemistry, so they proposed a third kingdom of prokaryotes that they call the eocytes. So they haven't really done that yet, really divided them into a different kingdom, but I guess people are continuing to talk about it. Okay, human relevance of these archaeans. The methane bacteria, okay, can be used for engine fuels, for creating methane for engine fuels, for heating, for cooking, and for light. Methane has an octane of 130. Okay, if you go to the gas station, those numbers that you see at the pump, whether it's 79 or 80, 84, 82, that's actually the octane. So methane has high octane. Okay, it's been used as a fuel in Italy for more than 40 years. Okay, it's clean, non-polluting, safe, non-toxic. It prolongs the life of engine, so this is actually a methane run engine here. This is like in a farm or rural community. Okay, so the bacteria will digest waste in the absence of oxygen gas. Okay, so 20 pounds of horse manure or 10 pounds of pig manure can be burned daily in a methane digester. That would generate enough gas that would be needed for an American's cooking needs for one day. Okay, if you were to use green plant material, less green plant material would be needed. Okay, so a digester is an airtight drum connected by a pipe to a storage tank, and there's a need to draw off the sludge, right, because you're actually using manure, right? So the sludge would, of course, make an excellent fertilizer. Okay, so here's a anaerobic digester. Here's a sludge zone. Here's a fluid. Here's where the gas is collected. So the pipe actually leads out of it from the gas, and here you have the fluids, also the effluents. Okay, so this is a look at methane digester. Okay. So France had more than 1,000 methane plants in the 1950s. They might have more now. This is just showing that the technology has been around for a long time for that. Okay, in India, cows produce more than 800 million tons of manure per year. Okay, so with the manure-fed methane digesters, many villages meet their fuel needs this way. And in the United States even, it's used on a small scale in rural areas. Okay, now viruses. Now smallpox is an infectious disease that had been widespread for thousands of years, killed countless millions of people probably. So here's actually the virus. Okay, it causes blister-like lesions which scar into pits. So in 1901, there was a little outbreak of smallpox and 720 people died. Here's a man with smallpox lesions. These are the scars from smallpox. Okay. So it's been eliminated in the United States by vaccination. So what they do is they introduce a weakened form of the disease agent or the smallpox virus into people through inoculations. I'm not even sure if they still inoculate people against smallpox in the United States anymore. Okay. So with vaccines, the body's immune system actually builds immunity. Okay. So the first vaccination happened in England. Okay, there were first vaccinations. Benjamin Jesse, a farmer, did one, and Edward Jenner, a physician, did another. Okay, they both saw that farmhands that worked with cows that had cowpox, they did not get smallpox. Okay, during epidemics that occurred periodically that kind of ravaged the countryside and the cities, right? So in 1796, Jenner scratched a boy with fluid from a cowpox blister on a milkmaid. Okay. And then 6 weeks later, he scratched the boy with fluid from a smallpox blister. And we might kind of consider the ethics of doing this, but the boy did not get smallpox. Okay. So Jenner performed a successful vaccination more than 50 years before Pasteur developed the germ theory of disease. Okay. And the word vaccine actually comes from the word vaca, which means cow. Okay. So people know that smallpox is caused by something smaller than a bacterium. So here's somebody with smallpox blisters on them. Looks terrible, doesn't it? Okay. Now during Pasteur's time, any infectious agents, whether they're bacteria or protozoans or yeast, etc., they were all called viruses. But there was a man named Charles Chamberlain who found out that porcelain filters block out bacteria, but that agents of the disease that could pass through the filters, they called filterable viruses. Okay. Later, all these different organisms that cause smallpox, rabies, measles, mumps, chickenpox, polio, yellow fever, influenza, fever blisters, warts, common cold, they are now referred to as viruses. Okay? Smallpox, rabies virus, measles, mumps virus, chickenpox, polio virus, yellow fever virus, influenza virus, cold sores or fever blisters, warts, and the common cold virus. Isn't that great, all these nice images? Yeah. Now, viruses lack cytoplasm and cellular structure. Okay. And so viruses may be the interface between biochemistry and life. They're sort of the interface between life and non-life. Okay. Now in 1946, Wendell Stanley won the Nobel Prize. What he did is he showed that a virus that caused tobacco mosaic could be isolated, purified, and crystallized. Okay. The crystals that were acquired that way could be stored indefinitely, and they always produce disease in healthy plants when the healthy plants were exposed to the viruses. Okay? Now, viruses do not grow or increase in size. Okay? They also do not divide. They do not respond to external stimulus. They cannot move on their own. They have no independent metabolism. But when they're inside living cells, they can express genes and produce more virus particles. Okay? Viruses are also incredibly numerous. Okay? In 1989, at the University of Bergen in Norway, marine biologists kind of figured out that 1 teaspoon of seawater has more than 1 billion viruses in it. Okay, so they're incredibly numerous. Okay? They're about the size of a large molecule, about 15 to 300 nanometers. Okay, so thousands of them can fit into one bacterium, and you remember how small bacteria are, right? They have a nucleic acid core, either DNA or RNA, which is surrounded by a protein coat. It looks like a little spaceship here. Okay, So they're classified according to their DNA or RNA. Then they're grouped according to their size and their shape. And then by the nature of their protein coats and by the number of structural units in their cores. Okay? Now, bacteriophages or phages are viruses that attack bacteria. Okay? As you can see, they look like a spaceship, like that other one we were looking at, like something in a sci-fi movie, right? They're made up of a head on a thin cylindrical core, okay, surrounded by a sheathing coat, okay? And at the base of the core, you have 6 spider-like fibril legs. Okay, so here's the head, here's a cylindrical core surrounded by the sheath, and here are the legs that help them actually anchor them. Okay? So viruses replicate at the expense of their host cell. So here's a bacterial cell, here's some viruses on it. Okay? First of all, they attach to the cell, then they penetrate. Some leave their coats outside the cell, and the DNA or RNA that they inject into the cell directs the synthesis of virus particles, and then they'll assemble into viruses into the cell. Then they're usually released as the host dies. Okay? So some viruses, like influenza, mutate very rapidly. They constantly need to keep up with new strains, with new vaccines. Okay? And some viruses greatly affect the metabolism of their host cells. So here's some bacterial cells that have been infected by viruses. You can see they don't look too hot, right? Anyway, so botulism bacteria can produce toxins only if specific phages are present and active. We talked about botulism before, right? Also, many forms of cancer are caused by viruses. So it's theorized, or people believe, that all living things have inactive viruses in cells. So the big question is, of course, what causes viruses to become active? Yeah, I don't know. Nobody knows. Okay, but could be chemicals, could be radiation, could be mechanical damage, right? Now, in higher animals, when they're invaded by viruses, they produce a substance called interferon. Okay, and this interferon is released into the intercellular fluid or into the bloodstream. Okay, small amounts of it come in contact with the cells, and that causes production of a protective protein, and this protective protein prevents or inhibits propagation of viruses in the protected cells and inhibits viruses from causing tumors. Okay? Now, because of these properties of interferon, okay, they like to produce large amounts of it, as much as possible, for possible uses against cancers and other virally caused diseases. Okay, so so interferon is a good thing, or it's like a treatment for cancers and other caused illnesses. Now, also in 1981, the University of Washington, along with Genentech, developed together. They said they were able to splice an interferon gene into yeasts, which are fungi. Okay, so they're larger cells, right? So they can produce more interferon at a faster rate and a lot more cheaply than bacteria do. Okay? Okay, now the human relevance of viruses. The economic impact worldwide is enormous. Okay? The common cold and flu loses millions of hours of lost work time. Okay? Infectious hepatitis still claims victims. Guillain-Barré syndrome and Epstein-Barr, okay, also infect people. And these are caused by viruses that are carried by almost everybody. Okay? So what actually triggers the active infection is unknown. Okay? So these actually affect the nervous system. Here's a neuron. Okay. Now, HIV is caused by a retrovirus that's related to those that cause cancer. And of course, they're looking into a vaccine that's in development right now. So vaccine production is a multi-million-dollar industry, and other viruses are used to infect ticks and insects and other disease organisms of plants and animals. There's a phage that attacks nitrogen-fixing bacteria in the roots of legumes, and other phages attack diphtheria and tuberculosis bacteria, which is what we would like, right? Now we come to another subject, which are the viroids and prions, okay? Now there are more than a dozen plant diseases caused by viroids, okay, so such as chrysanthemum stunt disease and potato spindle tuber disease. So the 3 potatoes on the left are victims of this potato spindle tuber disease, and the potato on the right is a healthy tuber, okay? So what are viroids? They're circular strands of RNA in the nuclei of infected plant cells, and they're transmitted via pollen or ovules or machinery. They can be seed transmitted too, okay? Now prions are particles of protein that cause diseases of animals. So this is actually brain tissue of animals that have been infected by prions. You see the spongiform, the spongy-like texture of it. It causes holes in the brain. Okay, so Creutzfeldt-Jakob disease and chronic wasting disease of deer, elk, and moose are actually caused by prions. Okay, they're thought to induce abnormal folding of proteins in the brain that leads to brain damage, and these diseases, these prion diseases, are always fatal. Okay. Anyway, so they also include scrapie and mad cow disease, things that you've probably heard of in the news that have killed, countless animals and also people too. Anyway, that's all we have time for today, and what we'll do is we'll see you next time. Okay, until then, aloha.